Okay, back at it again. So this will be part two of cells, kind of the big picture of what's going on. Um, so there are some basics about cells you need to know. For example, you should know the difference between prokaryotes and eukaryotes. All cells do have some parts in common. For example, cell membranes or plasma membrane, which are interchangeable. Cytosol, which is not cytoplasm. Cytosol is the liquidy portion of the cytoplasm. The cytoplasm includes the cytosol, the liquidy portion, plus all the things that are floating inside inside the cell from the inside of the membrane to inside of the membrane. DNA and ribosomes. So all living things need to have a way to separate themselves from the outside world, have some type of a liquid medium inside the cell. They have to have a genetic code that they pass on, and in some cases, the genetic code isn't DNA; it's actually RNA. But in general, DNA is the most common, and then ribosomes are used to make proteins. So there are we actually did this. The first lab we did had something to do with the limits on cell sizes. So cells are small because they need to be small. They need to have a really high surface area to volume ratio. So they have to be bigger on the outside than on the inside. We saw this with the cube lab that we did with the three cubes, large, medium, small. When we soaked them, these agar cubes, in different solutions, I think we used sodium hydroxide. The cubes change color from like kind of a bland color to a bright fuchsia or pink color. The one that was the smallest absorbed the most in the same amount of time as given to the large and the medium-sized cell. And because of that, that shows you that the surface area has to be small in order to feed the inside of the cell and also get rid of waste products. Couple thing we'll look at here. So here's an example of a prokaryotic cell. They have these. There's a cell membrane or a plasma membrane on the outside. Almost all bacteria or single cell things that are prokaryotes have some type of a cell wall. That cell wall it's used to give them shape, structure, rigidity. It's an anchor point for things like pili or cilia or flagella, which help for movement. And once again, obviously, they're going to have these ribosomes, and here's their kind of genetic material. They have this idea called the nucleoid region, which is just a darker spot within inside the cell that actually contains the majority of the genetic information. We talked about types of microscopes. This is a transmission electron microscope. It actually allows you to see inside or transmits through the membrane or through the cell wall of a particular type of cell. Go back real quick. So these cell walls, they have this protein called peptidoglycan. It could either be, this should be gram, not GRM. It should be gram positive or gram negative. One places you may have seen this is with mouthwash. So, for example, when you're a little kid and you're learning how to brush your teeth, one of the things they've come up with is this mouthwash that stains your teeth either green or red or blue or whatever. The idea is that mouthwash sticks to the cell wall of certain types of bacteria. So if you rinse your mouth out with this mouthwash, any places that you missed brushing that still have bacteria or plaque there, this chemical will stick to it. It sticks to its base on the gram negative or gram positive type of cell wall. Not super important for our level. It's something you might see later on through your time in science. If you took a course of microbiology, it's huge. If you've ever gone to the doctor and they do a throat swab, a culture, to see if you have strep throat, in addition to looking at the shape of the type of bacteria, for example, this is a bacilli-style bacteria or rod-shaped bacteria, they're also looking to see is it gram positive or gram negative. Depending on that, they'll then give you certain antibiotics. Eukaryotic cells, pretty straightforward. They have internal membranes. They have a system of these membranes. The big components that you're going to see are obviously a nucleus, but you'll see smooth ER, rough ER, Golgi, vesicles, vacuoles are all parts of these eukaryotic cells. In addition to that membrane system, you're also going to see things like chloroplast and mitochondria that have evolved to live in our own cells over the course of hundreds and probably maybe even billions of years in cell evolution. Typical animal cell. Not every animal cell will have every part you see here. Obviously, not every animal cell will look like this. This is the cell that you will be responsible for in any test and quiz we might have. I will give you the same exact diagram, the same picture. You should know what all these parts do. I'm not going to run through them now. We'll spend a little bit more time in class on each one. But if you know 50% already, you're in good shape. Here are some things that are not found in animal cells. Obviously, chloroplast, a water vacuole, or tonoplast, which is the surrounding of the water vacuole. Something called plasmodesmata, what we'll get to, and obviously a cell wall. Typical image of a plant cell. Once again, not all plant cells look like this, but if we kind of toggle back and forth, you'll see that in general, animal cells tend to have a less defined structure in some respects, all right, than the plant cells. Animal cells tend to be more diverse, so when you look at types of animal cells, you can actually determine. If you ever take a class in histology, you'll talk about the how certain cells look will dictate what their function is. So a couple major kind of takeaways here. There are these tiny little holes that go from cell to cell. These are plasmodesmata, super important in plants. So every single plant cell is connected through these holes with every single plant cell, which is a huge benefit for plants. It allows them to move water and nutrients across from cell to cell to cell. All right, so here's one cell over here. Here's the cell in question. Here's a third cell on this side. You can move water and nutrients from a cell that's over here through this cell to a cell on the other end super fast, super efficiently, without having to cross membranes. It's also a negative for plants. So, for example, if something that's small enough that gets into plants that maybe causes infection can also move from cell to cell to cell. Humans do not have, or actually eukaryotes, animal cells that are eukaryotic cells, do not have these plasmodesmata. They're only found in some places. The only place I can think of off the top of my head would be heart muscle cells have the same these things. They're called gap junctions, and the idea of the gap junctions in heart cells are to send electrical stimuli from one cell to the next to allow heart muscle to contract in a quick and effective manner. All right, I'm going to stop here. Once again, these are going to be short, quick little videos. You are responsible for all the pictures, all the parts here and here. You should also know what these parts do. You should know what parts are found in what types of cells, like what are found in plants, what are found in animals, and I hope this helps.